After studying this module, you shall be able to understand the concept of circadian rhythms and zeitgebers, learn the structure and function of the SCN, learn about other seasonal rhythms, learn the role of pineal gland in maintaining rhythms. Let's just start with an introduction first. Biological rhythms are cyclic changes in the way biological systems behave. These biological rhythms are cyclic behavior patterns of varying length displayed by animals, plants, and even single-celled organisms. Most of our behavior occurs in a rhythmic pattern. Because these rhythms last about a day, they are called circadian rhythms. Circadian rhythms refer to the 24-hour cycle which is endogenous. The word circadian comes from the Latin cicera, which means about, plus dies, which means about a day. The two best known circadian rhythms are the sleep-wake cycle and the body temperature cycle. Earlier research has indicated that humans may settle down into a cycle of roughly 25 hours when isolated from external stimuli like light and timekeeping. The biological rhythms are controlled by first internal biological clocks called endogenous pacemakers and second external cues from the environment called endogenous zeitgebers. These include sunlight, food, noise, or social interaction. The circadian rhythm is not the only biological rhythm. There are two other important rhythms, ultradian rhythm and infradian rhythm. The ultradian rhythm spans less than a day. There is a 90-minute cycle of slow wave and REM sleep. The 90-minute sleep cycle is itself located within a 24-hour circadian cycle, so it would make sense to find that this 90-minute clock was also clicking throughout the day. It is called the basic rest activity cycle. It can be abbreviated as BRAC, B-R-A-C. Kleitman found that eating, drinking, urination, heart rate, oxygen consumption, stomach motility, and performance on various physical and mental tasks followed a 90-minute cycle. A need to recharge oneself was felt after each cycle. Even the infants demanded to be fed after every 90 minutes. This 90-minute cycle is also known as ultradian rhythm and indicates heightened and lowered level of brain arousal. The infradian rhythm has a period of more than 1 day but less than 1 year, such as the menstrual cycle in women and seasonal affective disorder. Some monthly and even yearly rhythms, particularly in the context of hormonal changes and reproductive behavior, have been observed. Passing of seasons, weather patterns, tidal waves, rising and setting of sun, menstruation in females, even rhythms of heart rate and respiration are examples of cycles governing nature as well as living organisms. We'll now talk about the zeitgebers. The internal clock which controls circadian rhythm is reset by light. The shift of activity produced by a synchronizing stimulus is referred to as a phase shift, and the process of shifting is called entrainment. Any cue that organism uses to synchronize its activity with the environment is called a zeitgeber, German for the time giver. Light is an important and powerful zeitgeber and it can be manipulated in the laboratory. In an experiment by Grobarski, Nuns and Gold, the activity of rats was monitored by changing the level of illumination from normal day and night pattern to constant dim light. When the rats were kept in constant dim light, they exhibited a pattern of greater free running activity towards evening and midnight. However, when the lights were on from 7 p.m. to 7 a.m., the greater activity was observed in the morning and afternoon. Thus, the level of illumination, the zeitgeber, determined the cycle of rest and activity. We'll now talk about the neural basis of circadian rhythms. If the sleep-wake cycle is under the control of a circadian pacemaker, then where in the body does it exist? One of the first scientists to address this question was Carl Richter, who found that large lesions of the hypothalamus interferes with the circadian rhythms. He made lesions to many different brain structures and examined their effects on circadian patterns of locomotor activity of rats. Only one structure appeared to disrupt circadian behavior, the ventral hypothalamus. But partly because this region was so small and anatomically complex, he was unable to go further with his investigation. It was subsequently discovered that a tiny part of the hypothalamus, the suprachiasmatic nucleus, named for its location above the optic chiasma, served as a biological clock. Lesions confined to the SCN portion of the hypothalamus interfere with the circadian rhythms of drinking and locomotion and of hormone secretion. However, it was hypothesized that the SCN control the rhythm, but the time analysis for the activity is controlled by some other area of the brain. Now let's talk about the structure of the SCN. The suprachiasmatic nucleus, the main endogenous pacemaker, is a tiny cluster of nerve cells which lies in the hypothalamus. It is located just above the place where the optic nerves from each eye cross over, called the optic chiasm, thus supra means above the chiasm. In fact, SCN is a pair of structures, one in each hemisphere of the brain, and each of this is divided into a central and dorsal SCN. The ventral SCN is relatively quickly reset by external cues, whereas the dorsal SCN is much less affected by light. The latter is therefore more resistant to being reset. The structure of the SCN has been investigated in rat brains. It consists of approximately 10,000 neurons densely packed with each other. These neurons are extremely small in size. The unique feature of these neurons is that their dendrites synapse with each other. This kind of connectivity is perhaps responsible for the maintenance of rhythms. The structure of the SCN is shown in this figure. Another special feature of these neurons is that they contain a large amount of RER, which is the rough endoplasmic reticulum, which helps in maintaining the production of proteins. They secrete neuromodulators and are therefore neurosecretory cells. Such neuromodulators form the chemical base for the control of biological rhythms. Now an interesting question is, how can light be a zeitgeber for setting or even resetting the rhythm of rest and activity if the SCN is located so centrally in the brain? How does the light reach here to serve as a stimulus for the cycle? The studies have indicated that the light from the visual system does reach this small nucleus. There are two pathways that accomplish this. First, there is a direct projection of fibers from the retina to the SCN. It is known as the retinohypothalamic pathway. Cells located on the ventral and the medial areas of SCN receive such an input from the retina. Further, investigations have revealed that rods and cones, the normal receptors of light on the retina, are not involved in this process of sending information to the SCN. Even blind rats are found to be following the zeitgeber manipulated from outside. Friedman in year 1999 found that mutation of genes necessary for the production of rods and cones on retina did not destroy the circadian rhythms. Provencio 2000 discovered the photochemical and the receptive cells responsible for the rhythms. The chemical is known as melanopsin. The site of special cells projecting to SCN was found to be around amacrine and ganglion cells rather than rods and cones. It was thus found that there is a special mechanism for connection between light and the SCN. Earlier studies have shown that the FOS protein is produced in the cells when they are active. Experiment manipulation of the level of illumination was found to be related to the amount of FOS protein generated in SCN cells, showing that they do respond to light, and light does serve as the zeitgeber for the circadian rhythms. The neurotransmitter found in the SCN cells is glutamate. The second route of projection of light to SCN is the geniculo-hypothalamic pathway. The lateral geniculate nucleus of thalamus, which receives optic information from the retina, projects information to the SCN. This is an equally important pathway that functions in coordination with the retinohypothalamic projection. The routes of the two pathways divert near the optic chiasma. Thus, even though SCN is not a part of the visual system, the light reaches it through two different pathways. Light is thus a zeitgeber for maintenance of circadian rhythms. The connection between the visual system and the SCN is shown in this figure. Now we move on to the mode of functioning in the SCN. The clock-like nature of SCN is also evident in its metabolic activity. If we take SCN cells out of the brain and put them in a dish, their electrical activity remains synchronized to the light-dark cycle that the animal had previously experienced. This striking evidence supports the idea that the SCN contains an endogenous clock. But even stronger proof that the SCN generates a circadian rhythm comes from transplanting the SCN from one animal to another, which suggests that the mode of functioning of the SCN neurons is chemical rather than electrical and axonal. In a study by Lehman 1987, SCN was first lesioned and then transplanted. It was found that the circadian rhythms which were destroyed by the lesion got re-established through the process of transplantation. The efferent connections were, however, not re-established. In another investigation by Silverne in 1996, the SCN of hamsters was destroyed, which abolished their circadian rhythms. Then, small semi-permeable capsules were used to carry the SCN tissue taken from another animal. The capsules were transplanted into the ventricles of the experimental animals, where no fiber connections could be established since ventricles contain only the cerebrospinal fluids. The nutrients reached the SCN tissue through the semi-permeable covering, but the axons could not connect with each other. The importance of the chemical base of functioning was evident through this investigation. Loud noises and changes in temperature are other environmental factors that have been associated with circadian rhythms in terms of setting or resetting the cyclic durations. Now we will talk about the radio-graphical investigation of SCN. Schwarz and Gaynor injected rats with radioactive 2-deoxyglucose. This substance is similar to glucose, but it can neither be utilized by the cells nor can it leave the cell once it has penetrated them. So it gets accumulated in the cells. When viewed under the microscope, it shines because it is radioactively tagged. For microscopic examination, the animal has to be killed and the slices of the brain prepared for examination. In the above mentioned study, the amount of radioactivity indicating greater or smaller activity of the SCN cells could be clearly differentiated for day and night activity for the rats. Greater metabolic activity was seen during the day. How do cells in the SCN maintain the rhythm? Is the question that still needs to be answered. In an innovative experiment by Welch et al. In 1995, they removed the tissue from SCN of a rat. They put it into papain, an enzyme which dissolved connections between the cells. Thereafter, they placed cells on an array of microelectrodes so that their electrical activity could be measured. The cells independently became activated and showed circadian rhythms. The most striking finding was that peak activity occurred at different times in each cell. So each cell maintains its own rhythms and they do not fire in complete synchrony. The question is, if the cells do not have synaptic connections between them and they do not even have a common circadian rhythm, what is the basis of a synchronized control of the rhythm? What causes intracellular ticking and timing? The obvious hypothesis is that some chemical is responsible for this cycle. Such mechanisms were studied in fruit fly by Hunter, Renser, Osley and Sehgal in 1996 among others. They showed the activity of two genes, PER periodic and TIM timeless. At the beginning of the cycle, these genes became active and caused the production of their specific proteins. When the production of proteins reached a particular level, these genes turned off further production of their proteins whose levels soon began to decline. When the level had declined to a particular level, the genes became active again and initiated the next cycle. Ralph and Menaker in 1988 showed that circadian abnormality was due to the mutation of a gene called tau, TAU. Analysis of the molecular structure of this gene led to the discovery of circadian timing early in the history of evolution. Now we'll talk about the regulation of seasonal rhythms. Cells in the suprachiasmatic nucleus seem to be involved in rhythms beyond the 24-hour cycle also. One of the interesting experiments in this context was done by Rusak and Morin in 1976 on male hamsters. The hamsters show an annual rhythm of testosterone secretion and their reproductive season. Their breeding season coincides with the light present in the environment. The breeding season is active in summer and declines in winter when lesser amount of light is present. The results of the investigation indicated that lesion of SCN cells was linked with the loss of this annual rhythm, and the hamsters even started secreting testosterone all through the year. One of the major centers involved with the control of seasonal rhythms is the pineal gland. The pineal gland is a small structure like a pine cone and is located on the epithalamus. It is located on the midline of the brain, just ventral to the rear portion of the corpus callosum. The location of the gland is shown in this figure. Pineal gland is a part of the endocrine system and secretes a hormone called melatonin. Melatonin is synthesized from the neurotransmitter serotonin in the pineal gland. Secretion of melatonin is at the peak during the night and declines as the day advances. Melatonin has a role to play in sexual maturation in coordination with the light. It causes early maturation in the tropical regions with abundant light and a much slower one in cold countries. The chemical called melanin also causes darkening of the skin. Besides such endocrine functions, pineal gland has been found to be involved with the control of seasonal rhythms in animals. The SCN has an intricate circuit to reach the pineal gland. The SCN sends signals to the pineal gland, directing it to increase production of the hormone melatonin at night. Melatonin induces sleep by inhibiting the brain mechanisms that promote wakefulness. Neurons in the SCN make synaptic connections with an area of the hypothalamus called the paraventricular nucleus, the PVN. The neurons on PVN then extend excitation to the spinal cord and synapse with the preganglionic fibers of the sympathetic nervous system. The excitation then travels through the postganglionic fibers and stimulates the pineal gland. The connections between SCN and the pineal gland are established through the synaptic connection. So when SCN is destroyed and then transplanted again, the 24-hour cycle, the circadian rhythm, is re-established, but the seasonal rhythms which are mediated through the pineal gland fail to reappear. Several studies have shown that large doses of melatonin during the day, when levels of endogenous melatonin are low, lead to quicker and better sleep patterns. A dose of melatonin before dusk may help travelers suffering from jet lag while going towards east, since melatonin is associated with darkness and east experiences darkness much later than the west. Similar adjustments can be done for the travelers going towards the west. Now we'll talk about the clocks other than the circadian rhythms. Many organs and cells in the body maintain more or less independent circadian rhythms outside the SCN. It has been shown that other rhythms are retained, whereas the circadian rhythms are abolished by the bilateral lesion of the SCN. There are other clocks to maintain them. These clocks, called the peripheral oscillators, are found in the adrenal gland, esophagus, lungs, liver, pancreas, spleen, thymus and the skin. Olfactory bulb and the prostate have also shown oscillations when cultured. Also, liver cells are shown to respond to feeding rather than light on any other zeitgeber such as temperature. Green and Minacker in 2003 showed that even though most cells of the body contain a genetic circadian clock, they are normally entrained by the neural and hormonal signals from the SCN. We'll now talk about the biological rhythms and human health. Biological rhythms are driven by the endogenous oscillators. Some of the oscillators are easily reset by exogenous zeitgebers such as daylight, meal times and so on, whereas other oscillators are more resistant. The result is desynchronization. Health problems can result from a disturbance in the circadian rhythms. Disruption in the circadian rhythm is associated with obesity, diabetes, and other metabolic disorders. The two most common examples of the disruption of biological rhythms and the resultant desynchronization are shift work and aeroplane travel, resulting in shift lag and jet lag. Shift work and frequent jet lag have profound effect on health. Levels of alertness also decline due to that. It poses a risk for the cardiac health also in terms of hypertension and chronic inflammatory diseases. Fatigue, disorientation and insomnia have been noticed in pilots when they have long periods of awakening and crossing of time zones. Thus, there are enormous health, safety and economic benefits to figuring out how the circadian clock works. Now let's summarize the module. An organism's life is governed by cycles related to most of the functions. Circadian rhythms maintain timing related to day and night. The circadian rhythm is controlled by the suprachiasmatic nucleus, the SCN, located on the hypothalamus. Electrical and chemical activity of the cells on the SCN correlate with the activity levels during day and night in many organisms. The light reaching SCN via retinohypothalamic and the genico-hypothalamic pathway is the zeitgeber for cells on the SCN. The major mode of functioning of the cells of SCN is chemical. Lesion of fibers connecting SCN to nearby area does not disrupt the circadian rhythms. Individual neurons rather than the circuits or groups of neurons are responsible for keeping the time. Genes called TIM and PER are involved in maintaining the circadian rhythms. Molecular analysis of the gene tau, TAU, has shown the presence of circadian rhythms in organisms very early in the history of evolution. Pineal gland, in coordination with SCN, controls seasonal rhythms. Hormone of the pineal gland, melatonin, can help people suffering from jet lag or night shift work, etc. It has been indicated in several studies that several organs of the body contain rhythm and they oscillate at their own rates. Disruption in the circadian rhythms can lead to serious health problems. The connections between SCN and the pineal gland are established through the synaptic connection. So when SCN is destroyed and then transplanted again, the 24-hour cycle, the circadian rhythm, is re-established, but the seasonal rhythms which are mediated through the pineal gland fail to reappear. Several studies have shown that large doses of melatonin during the day, when levels of endogenous melatonin are low, lead to quicker and better sleep patterns. The clock-like nature of SCN is also evident in its metabolic activity. If we take SCN cells out of the brain and put them in a dish, their electrical activity remains synchronized to the light-dark cycle that the animal had previously experienced. This striking evidence supports the idea that the SCN contains an endogenous clock. Health problems can result from a disturbance in the circadian rhythms. Disruption in the circadian rhythm is associated with obesity, diabetes, and other metabolic disorders. The two most common examples of the disruption of biological rhythms and the resultant desynchronization are shift work and aeroplane travel, resulting in shift lag and jet lag. Shift work and frequent jet lag have profound effect on health. Levels of alertness also decline due to that.